Hi, Zhao.
Hi, Golden.
How are you doing?
Yeah, I'm great.
Good. So I've just been talking to Steve about all of this range of customised printers that we're making for our clinical collaborators. What's the clinical project that you're involved in?
This one is for the pancreas islet transplantation project. And this is the printer we developed for this project. We call it 3D PICT. It's the pancreas islet cell transplantation device. And in this device, we contain a three-dimensional printer with the core and shell structure. And this is some sample print in the core shell structure.
So the core and the shell, in this case, Steve was telling us about putting the cells in the middle of the core. What's the idea here? I remember that we need to integrate 3 different types of cells for the islet cell transplantation. So how would those 3 types of cells be integrated?
Yes, we encapsulate the pancreas islet cells in the core structure, which can provide more immunoprotection for the islet. And we can put the supporting cells such like endocellular progenitor cells and in the shell structure, and these cells will promote the vascularization process of the whole implant construct. So in the meantime, we also have this droplet device. They can drop the cells into the printed construct. So we have the potential to deliver 3 types of cells together into one construct, such as T-Rex cells, and these cells can protect the islet from the immunoprotection from the patient itself.
So the islet cells, they're obviously the key cell that the clinician wants to transplant into the patient. So they come from a donor, and they are going to respond to glucose and release insulin. And then you mentioned the endothelial progenitor cells, so they will help with vascularization, is that correct?
Yes. Yeah.
And then you mentioned T regulatory cells. Now, these T regulatory cells, as I understand it, they help to regulate the immune response, is that correct?
Yes.
Right. So this printer then can deliver all 3 types of those cells discretely in a 3D structure.
Yes.
Can it also spatially distribute them where you want them in order to optimize performance?
Yes, correctly. We can control the position of each type of cells. And for example, we can put the T-Rex cells in the surface, in the outer layer of the construct, and protect the islet cells inside the construct. And in the meantime, the EPC cells will go through whole construct and promote the vascularization of whole construct.
Great. And I know that this project is carried out very much in association with Professor Toby Coates' group in Adelaide. So how does that work? I mean, do they now have a printer in their laboratory?
We deliver a custom-made, is nearly identical to this one, to their lab. And they also work hardly on that device and try to print the construct for animal work. We already implant some construct in mice, then we are targeted to the large animal samples for such as pig. Yeah.
Right. So it must be exciting to be involved in a project where there's very much fundamental advances in materials, fundamental advances in how we understand the cells to work in combination with those materials, and then actually delivering those ink formulations in the hardware into a clinical environment where we can envisage translation of this technology to help individuals. That must be exciting.
Yeah, we are really excited to get involved in this project. We are not using our knowledge in the material and the biology and engineer, and to develop the device and formulate the bio-ink formulation, and which can be used in the clinical and have potential to treat type 1 diabetes.
Brilliant. That's great. Thanks, yeah. Thanks for providing those insights for us. Thank you.